Telomeres cap the ends of chromosomes, and their length determines the ability of the cell to divide.
Telomerase is an enzyme that lengthens telomeres on cells. When telomeres become too short, the cell experiences genetic instability and can no longer divide.
However, in cancer cells, overexpressed telomerase activity causes cells to keep proliferating and thus creates immortal cells.
We can trace this overactivity to the overexpression of the HTERT gene, the gene that codes for the catalytic subunit of telomerase.
The transcription factors c-MYC and HIF1 are responsible for the regulation of telomerase gene expression.
In this Western blot, gel electrophoresis was used to separate the proteins, and an antibody was used to stain for HTERT. HTERT messenger RNA is detected at elevated levels in cancer cell lines, as indicated by the darker bands.
Elevated HTERT means cancer cells have high levels of telomerase activity, which suggests that HTERT overexpression facilitates immortalization of cancer cells. Studies have shown that hypoxia can activate telomerase activity in cancer cells, allowing these cells to proliferate and cause disease. Hypoxia is a condition caused by insufficient oxygen delivery to cells. This is often the result of poor diffusion and a disruption in the circulation of blood near the affected cells.
Regions of hypoxia are found in most human tumors. Hypoxia is often linked to resistance to cancer therapy, increased genetic instability, and enhanced malignant tumors.
HIF1 is a transcription factor that regulates promoters containing hypoxia response element HRE sites, including the telomerase gene promoter HTERT.
The introduction of telomerase promoters with the HIF1 expression vector led to an increase in the activity of the active wild-type variant of HTERT. This increased activity suggests that hypoxia plays a role in regulating telomerase in tumors.
In addition to HIF1, c-MYC is a transcription factor that binds to the HTERT promoter region. In vitro studies have demonstrated how c-MYC binding increases HTERT activity.
Normal human mammary epithelial cells have no detectable levels of HTERT mRNA, but transduction with a c-MYC retrovirus leads to significantly higher levels of expression, as indicated by the darker bands in the northern analysis.
Similar results have been demonstrated in solid tumors, which have longer telomeres compared to normal cells.
Highly malignant round cell liposarcoma shows long telomeres and strong telomerase activity. Immunohistochemical staining demonstrates very high levels of c-MYC, as indicated by the red stain. Furthermore, HTERT expression is elevated in these cells, as indicated by the brown staining.
The transforming growth factor beta-1, TGF-beta-1, is shown to regulate telomerase activity through different independent mechanisms.
TGF-beta-1 interacts with c-MYC and causes the inhibition of HTERT transcription by phosphorylating and activating the inhibitory protein SMAD3. This protein migrates to the nucleus and binds to either c-MYC or the HTERT promoter site. It has been proposed that both interactions silence HTERT gene transcription, decreasing levels of telomerase in target cells. Further study of the TGF-beta1 and HTERT interface may lead to new anti-cancer therapeutics that target telomerase gene expression in solid tumors. For example, TGF-beta1 can be genetically engineered to increase SMAD3 phosphorylation in targeted cancer cells. We hope that better understanding of this process will improve cancer treatment in the near future.